Welcome back to lesson 5 of the basic Unix tutorials for the BVCN, working through the Happy Belly Bioinformatics Unix tutorials from Dr. Mike Lee. So we're here in our last example, our last tutorial step, number 5, called variables and loops, where we're going to use the information from, we're going to use Unix to work through and allow us to store things that we might be looking for, as well as allow us to process through multiple commands somewhat in an automated fashion. And so if you're following along in the binder and you are like me coming off of lesson number 4, you might need to move to our starting location using our absolute path to return to Unix_intro. So the first thing we're going to work with is how we're going to work with variables. And so it's a fairly easy format for this. So we come up with a name for our variable. In this case, it's called my_var. Again, we don't want to have a space here. If we were having a space, the Unix interpreter wouldn't be able to tell what we want. And we're going to call my_var. We're going to store in it the word Europa, or the moon Europa, I guess, but maybe not the whole moon. And so one of the things that we have now is in this instance that we're working in our terminal. So if we were to exit out here or to close this and start a new terminal, this would all disappear. This would not be stored here anymore. But for right now, anytime we want to interact with my_var, it's now going to be storing our term Europa. And so there are two ways that we can do this, right? We're going to use the word echo, or the command echo, which takes what you give it and just prints it out as in the standard out printing, right? So if we do echo and we use a $ to denote our variable, which is my_var, what will happen here is instead of just printing out what we gave it, it'll print out our variable. Where on the flip side, if we didn't use the $, it will print out it will just print out exactly what we gave it, which is how echo works. So here my_var. So you need that $ there to interpret the variable that you're giving. Not dissimilar to lesson 4 when we were using awk, and the columns were represented by a $, and the number of the column, which was telling it treat the entities in that column as its own instance, in that case as a variable as well. And so we can also store sentences in our variable, right? So we're going to create a new one called my_new variable, not new car, which would be fine, =, and because we want to give it a sentence this time, we're going to open quotes. Remember, spaces are special in Unix, so it would not be able to interpret this correctly without the open quotes. But with the quotes, we're saying this is a unit. Keep this together. When we hit enter, it will get stored. And then we want to do echo $, because we're going to treat this as a variable, my_new var. There you go. So the variable here is this whole sentence, and we're returning that as an element in itself. And so we can use that information. We can store things as variables going forward to make it so that we don't have to necessarily keep track of all of the things that we're putting into our Unix commands once we've done it the first time. So we're going to make a directory here using our make directory command, which we're going to call _for_loops. And if we hit ls, you can see that we have a new directory here in our Unix intro. And then we can cd into our for loops. We're going to make some fun commands here. And we're going to work with specifically 4 magic words, as Mike classifies them, that work in our loop structure. 4 is a magic word that starts our loop variable, right? So the Unix command knows what we're doing with. In is whatever we're looping over. Do is the thing that we want it to do. And then when we're done giving it things to do, you have the command done, which tells it to stop. And so we're going to use echo here. What we're going to do is we're going to type this out. So it's for item, right? So the thing that we're looking for in this set of components that we're giving, 3 words that we're giving, which is car, truck, ukulele. We're going to say do, and then we're going to say echo that item, right? So this item will step through these 3 terms and then print them to our screen using echo. And then when we're done, we'll hit done. And so we do all this in multiple steps, and you'll see what happens here. For item in car, truck, ukulele. Why would you pick ukulele as a word? I don't know how to spell ukulele normally. Okay, so we're gonna hit enter here. And so you're gonna notice that this changed. So what happened is by using for, we've now designated to the Unix command line that what we're actually gonna do is we're gonna work in this for loop for a little bit. And so instead of returning what we're used to here, which is the location of where we're working in as the shell, it's saying, okay, I'm waiting for your next step. What do you need me to do as part of this for loop? And so what we are gonna say is, this is what you are gonna do. You're going to do, let me make sure. We're going to put a space here. We're going to do echo $sign item. We're going to hit enter, and then we're going to hit done. And so this, when we're done with that, should create our loop for us. There you go. And so what it's done is it's gone through and each item in our lists of items, car, truck, ukulele, we go in, we're going to echo each of those items to the line and print them out, and then we're going to hit done. So if we had done this, if we had said for item in car, and we get into this point where we're in our for loop, but we didn't mean to hit enter yet, we can actually use Control-C to kick us out of the loop before we complete it. So there's other formats that we can do this in. And so this is one of those things that, again, that's always interesting to see how different people do this. So this would be how I would write something like this, where we actually all do it on one line. And instead of using the enters to denote where our next information is, you can use semicolons to say where the next step is. So this will do exactly the same thing that we just did in car, truck, ukulele. We're going to put a semicolon there. We're going to say do a thing, just echo item. And then when we're done, we're going to close it with a semicolon there. And it does the exact same output. So you know, again, approaching Unix in multiple and bioinformatics in general from multiple angles it's pretty hard, pretty easy to just come up with new ways of working through getting to the same solution using different tool sets. Okay, so here we're going to add a new line to what we're going to do. And so this might look familiar from the previous lessons, where we're going to take echo the item, which normally will print this to our screen. And instead, we're going to take the item and we're going to append it into a file that we're going to create called words.txt. So it's going to be very similar to what we did. We're going to do for item in car, truck, and then ukulele, spelled correctly, which will give us our prompts here to do this correctly. We're going to do, hit enter. We'll put a space or two spaces to make sure that it looks clean. We do echo item. And then our second command, which will also do two spaces, we'll do echo item, $sign item. And here we're going to append it to a file called words.txt. And then we're going to hit done. And so not only will we print our items to the screen like we did here, if we hit ls, we'll see that there's now a document called words.txt, which if we look into, we'll have our 3 words printed there. Okay, so one of the things that we want to emphasize going forward is why we used the double carets here, right, and not the single caret. And so if you wanted to do this, we could go through. Let's see. So you can see automatically if I'm down here at the bottom. If I start going back up through the previous commands, so here's the last one. Here's the one before that by hitting the up button. And then this one, you can see that Unix by default changes our input that we had with these prompts and gives it back to us as these in the semicolon format. And so I'm just going to use this version just because it's easier than retyping out. But if I go here and change it from 1 caret, or from 2 carets to 1 caret, and then hit enter. So we still get the same output to the screen. We do echo items because that was the first command that we did. I'll point to it here. But now instead of appending it to words, what we're actually doing if we do this is that we're actually just, every time we do it, we're overwriting the words.txt document. So the first time we do it, we create a document that has the word car in it, and then we overwrite it with the next one, truck, and then we overwrite it with the word ukulele. So that's not ideal if we're trying to keep it all. So I'm going to replace it. I'm actually going to have to remove words.text and then replace the words that we get so we get the right one that we want. So again, if I do head words.text, we get the 3 that were in there. And then Mike has this challenge to us of, can you think of where we would put a single caret if we didn't want to overwrite the file with each iteration of the loop? And this was I thought this was a very interesting question. And it was also one that I wasn't, when I saw it, I was like, oh, that is a good point. Where would we want to do this? The solution to the answer is that when we're working with this, we're asking it to echo out items to the text, to the screen, right? So that all comes out as a one instance after this whole loop is done running. So what we could do is we could rerun this part here for item in car, truck, ukulele. Man, I'm gonna be so good at spelling ukulele. Do, and then here we're gonna do 2 spaces echo $item. And now we don't actually need this second one, because what we're gonna do is we're gonna say done, and then after done, put it into our document words.txt. So this is gonna take our output, which is normally printed to the screen, and put it into words.txt instead of appending it, and instead of overwriting it, it's just gonna take all of our content from the end of the command and put it into the document that we want. So now if we do head words, you notice that it didn't print to the screen. That's because it got saved in the words.txt file. And so now what we're gonna do is we're gonna generate loops to use. Instead of giving it a list of items to go through, we're gonna use our document that we created as our source of that list. And so we can do that with a certain set of formatting. The one thing that we need to do to format this correctly is use the command cat, which is an abbreviation for concatenate. And so that's interesting. One of the things that's interesting here is, so cat, if you're using it, I'm gonna hit clear here so we can start from the top. We can use cat in a couple of ways. The main way that it gets used in a lot of instances is to concatenate things. You can take 2 files, file 1 and file 2. You can do cat file 1, file 2, and when you hit enter, it takes both files and it just sticks them together with a new line in between the 2 files. So it's the opposite of paste. If paste takes columns and sticks them together, cat takes 2 files and puts them on top of each other. The other thing that cat is also used for is to take a file that you're working with and bring it into the active part of Unix, right? So when I say cat words.txt, it reads it into active memory of the computer. So this does exactly what head does, but if the file happened to be longer, it would actually produce the whole thing. And so cat is also a shortcut for just bringing in a file to be in active memory. And so that's why we use it in this formatting going forward. So it's saying here, for each of the words, or for each component inside word.text, treat it as a variable, which is why we have that $. And we can do that by saying cat, read each component of words.text into the active memory of the computer. So we can do for item in, and here we're gonna do a $, open parentheses, cat words.text, close parentheses. So this is the same format that we were doing before, but instead of writing out car, truck, ukulele, we're looking at it inside the file that we stored them in. We can do, we put in 2 spaces, echo, space, $item, done. And that gives us the command that we've been working with this whole time, which is printing out the 3 names of those items as we work with. We can also use echo to see that this would be the same result if we typed out without doing the for loop. So again, lots of ways to get the same information that we are working with in many different ways. And so you might ask, well, if we can just do this with echo, why do we do it with the for loop in the first place? And this is, well, part of the reason is that it doesn't return on a new line. It just reads in the words the way that we wanted. And the other thing is that we can replace this echo item line with anything that we want, really, right? It can be a calculation. It can be another series of commands. So we're using echo, but it could be a grep command. It could be a paste command. It could be a cut command. And it could be a bioinformatics command that we get to later in these types of lessons. And so it leaves us at that power, right? So even though even though it didn't exactly do what we wanted it to do, which is interesting, but mostly it's also because in that for loop, we can actually put in other things that aren't just doing what we're asking it to do in one simple line. So this is classified by Mike as a bonus round, and it says interleaving files with paste. But really what we're going to do is we're going to set up through a series of commands that are going to use a lot, and I'm going to reemphasize this, a lot of the tools from the first 4 lessons to basically create new files, and then at the end of it, practice going through loops. And so this is going to be multiple steps that we're trying to get to. We're going to be using first we're going to use the command paste. And the target here is to initially make a file that represents a FASTA file. And so if that's something that you're unfamiliar with because you're not planning on using Unix for biology, FASTA files are a common file format that store a nucleotide and amino acid sequences. And it's a fairly simple structure that looks like this, where you have a header line with a caret and some information about what the name of the sequences that you're working with. Then you have a return character, and then you have the information of in this case, a DNA sequence, another return character, and then to denote that the next sequence is starting, you use another caret with another new name, and then more information about the new sequence there. And so this is going to involve a number of steps, like I said, using commands from the last 4 lessons. And so in this instance, I'm going to hit clear so we can have a clear page to start working with. So the first thing what we want to do is we're going to use our absolute position in our computer to copy from a different directory, specifically Unix_intro, from the 6 commands. There's a file that we were working with previously in other lessons called genes_and seeks.tsv. And so we're going to copy that, and we're going to copy it here by using this period to say this directory, copy this file to this directory. So if we hit enter, we're in our for loops directory, we hit ls, we now have this file present here. We can check to see what the contents of it are with head n-1 genes_and seeks. Okay, so this has 3 components to it. It has a gene ID, the amino acid length, and the sequence length, which is more information than we need to put into a FASTA file. So we're going to combine a number of elements here to create 2 different files to work with. The first file is going to be our IDs, so it's going to be the information from our first column, and then the second file is going to be our sequences, which is going to be the information from our third column. And then here we're going to actually use a nifty little thing about tail, which is going to say, hey tail, what you've seen previously, we've done both head and tail where we can say how many lines we want it to print for us. Here we're going to denote and say, hey tail, the number of lines we want is exactly all of the lines except for the header line of the table. And so we're going to denote that by saying +2, right? So it's saying, we want you to capture everything in this file. Here's the header, here's the end. Capture everything until you get to the top. And then we only want to get everything that starts at line 2 and nothing in line 1, which is good. So that means that we won't have this information. We'll only have the sequence information that we want. So again, we're preserving the order here, which is important because like we mentioned in lesson 4, there is no preservation there's no matching of information in using paste. So if you're going to use paste, you want to have exactly what you want in exactly the order that you want it to. So let's use these commands. We're going to do cut f-f1 genes, and then we're going to pipe it to tail. We're going to do -n. Here we're going to write +2, and then we're going to output it into a file called ids.tmp for temp. And then we're going to do the same thing. We're going to do cut -f3 genes, pipe it to tail -n +2. So almost exactly the same, except now we're capturing a different column and we're putting into a different file called seeks.tmp. And if we look at that information, we can look at IDs using head. It's just our IDs counting upwards, no header line. And if we do head seeks, it's going to be a lot of information about the amino acid sequences that are present there. Okay, great. So we're part of the way there. The other thing that we need to do is we need to convert our headers or our identifiers into header lines for a FASTA file, which involves like it says here, it involves adding this caret. And so we're going to use sed, like we learned from lesson 4, to replace. And so here we're going to say sed, anchoring at the start of each line, which is what this up arrow means, this caret, up caret. Replace that non-entity, that non-space, with a caret here to denote that it's going to be the start of a line in a FASTA file. So we can do that here. We're going to do sed. We're going to do our open single quote substitute. So at the start of the line where there normally is nothing, replace that nothing with a caret. And because there's only one start of each line, we don't have to say that this is global. We're going to do our IDs.tmp, and we're going to now put it into something called FASTA_IDs.tmp. And so we can compare these two files. And we can see that it's the same information as IDs.temp, but now we've added a caret into it, which is great. And so what we want to do now is we want to interleave our files together using paste, right? So interleave meaning we want the first line in the first file followed by the first line in the second file. And so Mike has a couple examples here of how not to do it. And so the first thing we can do, we can say, okay, paste. We're going to use paste. That's great. We're going to take our FASTA IDs. We're going to take our seeks sequences in our seeks.tmp file. We're going to pipe it just to see what this output would look like from head-n2. And, well, that's not great. That's not what we want. This isn't a return line. What we've done here is we've taken our identifier in our header line from our FASTA file. And instead of putting a new line, we've put a tab. And that makes sense because paste by default uses a tab as its a delimiter here. And so Mike says, okay, well, we can test this out in a couple of different ways, right? We could say paste. Let's change our delimiter, very similar to the way that we change our delimiter in cut with a -d. And instead of tab, we're going to do open double quotes. We're going to put a dash, close double quotes. We're going to use the same two files, our FASTA's IDs TMP and our seeks TMP file. We're again going to take a look at the output using head. And so again, not what we're targeting here, but we can see that we've manipulated the space here. So instead of being a tab, we've replaced it with a dash. Okay, so we've done all these nice fun rigmaroles. Let's change this now to be looking at a new line, right? Which is the next step here. So we're going to replace that dash with a new line. And then we're actually also going to change our head number so we can see the first two full entries from a 2 to a 4. And so we can do this using that up command, right? So if we hit the up button on our keypad, we can bring up our last command that we used and make just the small changes that we need to this previous command. So we can change this 2 to a 4. And I can change this dash to a representative of a new line, which is that \n. And then if I hit enter, so this is exactly what we want here now, right? We now have our commands, or we now have our output in our, we now have taken our FASTA IDs and our seeks, and we've now given them in our own line. So we have our identifier representing a FASTA identifier with a new line. We have our protein sequence, which then has a new line, and then the next sequence here. So again, we want to actually store this information and not just have it as our in head as an output. So I'm going to hit the up button again, and now I'm going to delete everything up to that pipe and replace it with a redirect to read it into a new file. That what I'm calling genes.faa. Now we can hit enter. We can take a look at our contents of genes.faa, which look very similar to this, and we're going to print the first 5 outputs. So here we go. Amino acid 1, 2, 3, 4, and 5. And so now that we're all done with this, we can look in here. We have our genes.fa file, but then we have a number of temp files that we started with that we no longer want to work with. So we can remove those all at once using remove our wildcard from lesson 2 that says anything that matches up to the point .tmp. And that will remove those files and leave us with just the final FASTA file that we wanted to work with. Okay, so now imagine we wanted to pull sequences that were the correct annotation for this. Ooh, this is going to get very fun and exciting here. So what we want to do is, in this instance, is we want to use the information that we have about functions that we're interested in. They're the genes that have been annotated a certain way. We want to figure out which gene that is, and then we want to just get the information from that gene from our FASTA file. So we're going to do that in a couple of steps. And so the first step that we're going to do is we're going to look for the gene identifier in our file that we were working with in lesson 4, gene annotations.tsv. We're going to cut out just that first field. So we're only interested in what is the number that matches this identifier, this KO value right here. And so you can see here that instead of using our absolute path, we're using our relative path. So this is saying go up one level of our directories, and then go, if it exists, which it should because this is where we are, if there is a directory called 6 commands, go into that directory, look into this file, run grep on this file, and then give me out that one output that we're targeting, or in this case, it'll be 2 outputs that we're targeting, and give me what that information is. So we can do grep, open quotes. Remember, so this is different than in lesson 4 where we didn't use quotes. So the quotes are just an optional element, except for the one instance which will show below why it's not optional. But so it can be a good habit to get into using those quotes, but it isn't necessary. So we're saying look up one directory, 6 commands, which I tabbed out because once we looked up one directory, we could see what else was there. Gene annotations.tsv, and then we're going to say cut -F1. Well, actually, let's before we do cut F1, let's just see what we would get back out. So this would be our regular output, which is the 4 columns that started in that gene annotations file. So when we say cut F1, we're only going to capture these 2 identifiers that we're interested in. So I'm going to hit the up button. I'm going to pipe it to cut -F1. You'll see that all we get back is that 7 and 67. And I can put those into a file that we're going to work with later called target_gene.ids.txt. And so now if we ls, so we were working with a file in the 6 commands directory, but then when we redirected it to a file called target_genes.ids.txt, it actually gets stored in the directory that we're working in. So when we hit ls, we can see it's now part of the directory that we're in. And if we hit head of that target file, you'll see that it matches just that head or that cut output that we were using just a second ago. So it's the 2 numbers that we want to work with. So we can look at this in a couple of ways, right? So the example is, so our goal will be to capture 7 and 67, but let's look at a couple of examples of how we would actually do this using the number 99 as our example here. So if we do, I'm going to hit clear just to give us some room to work with. So our genes, right? So we can take a look at this, our genes.faa file. So this contains all of the 100 sequences from the starting file here. It counts up, it gives us it in FASTA format. I use less, so if I want to get out of less, I hit Q. So if I do grep, and I say grep 99, so that would be the identifier number on the caret line. That's the header line for the FASTA from genes.faa. Okay, great. 99 is in the file, which we expect because it counts up to 100, but it only can capture for us the line that it's on. And the line that it's on is the header line that we work with. But grep has, as we see, a lot of these Unix commands have a lot of different parameters, and grep has a parameter that can help us with this. So if we do grep, and in this case we do capital A being important because lowercase a is also a flag for grep, 99, here genes.faa, and hit enter. Now not only are we capturing the line that it matches, but we're saying also capture the next line. So the line after the one that we're working on. So now we're actually capturing the line that has the information that we would want to go along with this. So it'd be interesting, we can actually do this. So what happens if we're like, okay, we want 99, but we also want the number 9 as well. So if we do that with grep A, 1, and instead of 99, we look for 9 and do genes FAA, you might be able to guess here that it's not going to just find the number 9, but it's going to find all numbers that have a 9 in it. And so this could get a little messy. So you can see we capture everything. We capture we capture 9, we capture 19, we capture 29, we capture 39, 49, 79, 92, 97, 98, 99. Obviously not ideal if what we really want to do is just catch the number, the identifier number 9. So we can actually use grep, a second flag that we're going to use here, which is a W. And the W stands for word. And so in this instance, it means a string of characters. So it could be numbers, it can be letters, it can be alphanumeric symbols that are recognized by Unix. And it can say, treat things as a unit if there's white space around them. Okay, so that'll allow us to say directly, okay, what we want is not 89 or 90, what we want is the word 9. So it's something that has white space before, white space after. In this instance, we don't have white space before, we have a caret, right? But that's fine because we can tell it, look for the caret as well. So it'll say, look for a caret and 9 together, treat that as a word, and only return things back to me that then are one unit like that. That the next element after the 9 is white space and not another number or some other element that might be part of it. So if we do that here, I'm going to clear just to give us more space. We can do grep-w, treat it as a word, give us 2 lines, the line that we match and the line after. We're going to search for the caret and 9, and then we're going to say genes.faa. And there we go. That's exactly what we want there. We're targeting the gene that we wanted and getting back this full sequence along with it. So if we wanted to, we could actually take a quick look at how this would look if we dropped the W. So this is going to be that intermediate step, right? So before, up here, we were looking at 9 by itself, and we got all of those options. We got 19, we got 90. If we did it like this and remove the word parameter, what we'll actually capture is all the instances where you only have caret and 9 together. So what we'll get is 9 and then all of the 90s entries as well. Just an interesting way that we can also look at that. So lastly, we're going to combine a lot of this information into one final series of loops that'll give us some information about how we're going to automate the process of getting the information that we want from a FASTA file in these loops, right? And so again, we're only working with 2, and that's fine. But you can imagine if we were working with a FASTA file that had 10,000 entries in it, and we wanted to capture 500 of them, we could list those 500 identifiers, which might be pretty easy to do, that we're targeting, give it to the loop, and the loop can process through all that information, give us out what we're looking for. Okay, so let's look at this way. So we have our file that we can do head on of target genes. There's 7 and 67. And so we're going to set up our loop like we have previously. We're going to say for gene. So that's just, we've placed the variable here. Instead of item, it's going to be gene. In our file that we're going to go through step by step using cat by reading it into active memory. $, open parentheses, cat, target, gene IDs, close parentheses. We'll hit enter. We'll hit do. We'll put 2 spaces, and then we're going to give it 2 commands. We're going to say echo gene, and then we're also going to give it 2 spaces. We're going to say echo, in this case, we're going to say print out the forward quote, or the forward arrow, the greater than arrow, our variable that we're working with gene, and then we're going to go to done. And so it's one of the things that's really important here to note, and one of the reasons why I mentioned when we're moving into working with grep, one of the things that might be useful is using these double quotes to note what you're looking for. Is this case example, edge case example of when we want to look for the caret. So when we want to look for the caret, if we were to do this exact code and then not have these quotes around it, what we would be telling it is echo redirect this input into a file called the variable that we're working for. So we would take the number 6 and we would put it in a file called 6. And so by putting the quotes around it, we tell it, okay, don't treat the caret as a redirect, treat it as an actual character in a piece of text. And so that works in the same way with grep. So we can grep the caret and target the caret exclusively in various ways as well. So in this instance, what we're going to do is we're going to echo out what's just the name, what is in the file. So we're going to echo out the gene as it is. So it'll be 7 and 67. And then we're also going to echo out 7 and 67, but with the caret in at the beginning of the line. And so if we hit enter, we get two outputs. So we get our 7 and then 7 caret, and then 67 and our 67 caret. And that makes sense because we're reading each line individually. So we go 7, 7 comes out as a gene. We echo 7 by itself, and then we echo 7 with a caret in front of it. And so we get 7 and then caret 7 and 67 and caret 67. And so we're going to take all this information. We're going to go and we're going to combine all of it. So we're going to use our loop structure to read through the file to generate an output. We're going to use our grep command from lesson 4. We're going to use our flags W and A to capture just specifically the number that we want. So specifically the number 7, no other instances of it. Specifically the number 67, no other instances of it. We're going to capture the second line so that we get the sequence with it. And we're going to pull it from, we're using grep here. So we're going to use all this information to pull it from a different file than what we're parsing through in our for loop. And we're going to read out all that final information at one time into a file called target genes. And so this is that output that we talked about before. One other way we could be doing this, right, is we could be appending it to our target genes, target underscore genes. We could do grep and then put the append sign here. Or we can do it all at once and output it as one final file. So this is our last loop for the lesson and for the tutorial. So we're going to do for gene in $, parentheses cat, and then we're going to do target gene IDs, right? So we have all of our information. This is what we're going to loop through. We're going to say do, and we're going to hit enter. We're going to put 2 spaces here to make sure that we are just have the formatting correctly so we can see what we're doing. So we're going to say grep. We're going to do -w-a1, look for all instances where there's a caret and the gene exactly as a word, so there's white space after it. We're going to do it from our file called genes.faa. We're going to hit enter. And then this is all done. We want you to put it into a file that we're going to call targetgenes.faa. We're going to hit enter. And now we're going to hit ls. We'll see there's a new file that we've added, targetgenes.faa. And if we hit head, targetgenes.faa, right? We took our target gene IDs, we parsed through our file, and then we generated a FASTA file that only has the 2 sequences that we're interested in. So that concludes our tutorial. This gives you an idea of how you might be able to use those loops as a powerful tool to parse through large files for specific elements that you want. We can combine it with our other tools. So here we combined it with our Unix commands, but we can do that going forward also with our bioinformatics programs that we work with going forward. And so with that, thank you for joining us. If you stumbled across lesson 5 without knowing where this started, lessons 1, 2, 3, and 4 are linked below in the description. This is all part of the Bioinformatics Virtual Coordination Network that's happening during the COVID-19 quarantine lockdown. And thanks for joining us.